All right, we're at the top of the hour here, so I'm going to begin. Welcome everyone to our Send Ashore webinar. We're excited and thankful to have you online today. If you have any questions during the webinar, we ask that you use the chat function or the Q&A section of this call. We will be posting the questions and answers early next week, along with the recording of this webinar. PointCross was founded in 1999, serving the life sciences industry since 2009. We participate in CDISC, Fuse, and within industry conversation with the FDA regularly, including as a contracting partner with the FDA. Today, we'll be talking about our Send Ashore service, which is part of our data standardization or DS services for non-clinical studies. Our DS services team also serve for send generation, interim study monitoring, study repository, all of which are done using our XBiome platform. Both large sponsors and small biotechs alike have entrusted PointCross with their non-clinical studies and clinical information, with almost 10,000 studies processed. PointCross's views are that the purpose of Send as both an analysis and submission artifact influences that our Send must be inclusive of all studied data. We believe that data types that require custom domains should be included in the Send data set. We also believe that compliance should not equal cost. We are committed to offering the industry's best value with our automatic send quote generator. If you visit our website, our homepage displays the automatic send quote generator as the first button, and that is this button at the top where you can just click automatic send quote generator. And it'll come up with a form that you can fill out. The generator includes both send generation as well as the send ashore process that we're going to be talking about in detail today. To fill out this form, all you have to do is submit a few details about your study, such as the study type, the link of the study, and how many subjects are within the study. What we're going to cover today are the challenges around send conformance, what is our send ashore process, and assuring reliability for your study. So if you are new to Send, Send is the standard for the exchange of non-clinical data, and it is a format mandated for submissions to CBER and CDIR for a variety of studies. If you don't know if your study qualifies for the Send requirement, please contact us. We are typically able to determine your Send qualification in just a few moments with the study title and the submission type. The Send implementation guide establishes a standardized format with a fairly strict set of rules. Currently, version 3.1.1 or IG 3.1.1 is the most up-to-date, but we are waiting the formal release and acceptance of the implementation guide 4.0. PointCross already generates custom domains for data that will be in the new 4.0 implementation guide, including ADA and immunogenicity data. In addition to the implementation guide, there are a number of other rules to follow with Send, and those are including but potentially not limited to the FDA business rules, FDA validator rules, FDA technical rejection criteria, PMDA validation rules, CDISC conformance rules, and CDISC defined conformance rules. One thing to note, the asterisk after the PMDA validation rules, PMDA currently does not require Send, but it does require electronic submission. They do, however, produce validation rules to provide guidance for your submission. There is guidance around the define.xml as well as SDTM, and SDTM rules are typically adapted or implemented as is for the validation of Send. I have here a quote from the FDA Technical Conformance Guide. Study data validation helps to ensure that the study data are compliant, useful, and will support meaningful review and analysis. As I mentioned, that is found in the Technical Conformance Guide, Section 8. This is later expanded upon in section 8.1.3 of the Technical Conformance Guide. Each study dataset must be 100% traceable to the final study report. Now, we know that reviewing every data point for both conformance and correctness does take time. Many of your send providers default to doing a spot check. We've seen this be as few as 2 subjects per domain. And if you're given a standard repeat dose toxicity study or and you're only doing 2 subjects, that can be as little as about 1% of the domain that is being reviewed, or a carcinogenicity study. If you're only doing 2 subjects or even 2 subjects per group, it can be as little as 0.5% of the actual data points that are being checked. If we are only checking a few subjects that are being reviewed, there are going to be plenty of areas for data points to be incorrect, and these are data points that are later used in important calculations. They could be missing due to a deviation. There could be data points that are out of range or observations with comments that are entirely missed. These would maybe not appear in the send data set, but they would be in the report. Additionally, there could be data entry errors that are easily entered from typographical errors, either during the initial data collection or during your send transformation. I've here another quote. It's coming from that same section 8.1.3. Based upon reviewer experience, establishing traceability is one of the most problematic issues associated with any data conversion. With that, I would like to introduce to you the Send Assure. Send Assure is PointCross's solution to FDA comments surrounding traceability, and Send Assure is an enhanced QC check. This QC check is going to be very in-depth detailed. It checks against the final PDF study report or a draft report if that's all you have available. Any study metadata, tabulated study data, validation and conformance rules that have been released, as well as the implementation guide's best practices, and any other guidance that may be released. And as I mentioned before, we are involved in these industry conversations. So some of those conversations do lead to some best practices that may not be implemented just yet, but are well accepted within the industry. This service is performed after your send creation and before submitting to an agency, whatever regulatory agency that may be. And the process includes a full reconciliation. Every data point that you are going to be submitting within your send dataset is checked. With that, what we can do is either correct the send data sets with any information that you are providing, or we can provide a log for the original send preparer. If you happen to be a send preparer and just wish to have an extra check, we would be able to provide that to you. Or if you would like us to correct that for you, we would also be able to do that. Important to note that 100% of our reviewed studies had issues. Now, of course, that's a bit of a shock. It is not 100% have rejection issues, but we have identified issues that would have impacted the review. So they may not have been technical rejection criteria. Those I think most of industry has down, but the data set may have been incomplete in some way or misrepresented the data between the report and the send data set somewhere. Some of the common examples that we have these findings, we may ask that TS parameters, your trial summary parameters, are present in the report, that they be added, maybe using a more recent control terminology or implementation guide based on when the study and send were performed. There are examples of modeling more effectively, such as using one washout period without a duration, as opposed to a 1-day washout and a 2-day washout and a 3-day washout period. Sometimes it's also including data that would require custom domains. As mentioned, we do include custom domains for things such as your ADA or immunogenicity or even the gene tox, which up until 2 months ago was not exactly accepted, but now it is. And then we would also have the missing fasting or baseline flags. Those are pretty common to be missed. Um, if they're indicated in the report, we do believe that they should be included. Now, I mentioned that there are two different ways of going about the send ashore deliverables. On the left-hand side is the traditional service. And then on the right-hand side, we have the optional service on top of that, which is with corrections. So along the left-hand side with the traditional service, your deliverables are going to include the aid data validator reports. So there's going to be two for the data set itself. These include the FDA and CDISC rules, as well as a separate one for PMDA rules. And then with the define XML validation report, we also have two separate ones for the FDA rules and the PMDA rules. We also then have the quantitative and qualitative reconciliation report, and that is going to be a 100% check of your send data set against the final report and the individual data points in there, as well as the send ashore report, and I will give you more information on that in a moment. On the right-hand side, we do offer optional corrections with that. So it includes all of the deliverables from the traditional service, as well as all corrections that are identified will be fixed in your send dataset. This will include all of the XPT files that you would be expecting, the NSDRG file, and the define.xml file. This might be an option for you if your original send preparer no longer offers the service, or if your organization has acquired a previously studied asset and you are looking to make them a little bit more consistent. Getting into some of the details, I would like to describe a little bit more about the eData validator reports and what that includes. So our eData validator are coming from, or these EDV reports, as you will see mentioned, are coming from the PointCross eData validator software. It is not just applicable to send, we do have SDTM rule sets populated within there. And the validator is populated with multiple rule sets and control terminologies. So these are going to be your FDA business rules, CDISC conformance rules, as I mentioned, the PMDA rules. Those are all going to be included, as well as every control terminology that has been released. Our recommendation would be to use the most recent one as to when your send was prepared. eData Validator also lists a number of PointCross specific rules. So we have rules in there such as consistency between significant digits for a single test. So for example, in the PC domain, all of the significant digits will be checked for consistency across there. The study is going to be run through the program, and it automatically produces a list of all of the errors. These could be errors, warnings, flags, whatever you'd like to call them. There are some severities to them. Things such as technical rejection is obviously the most severe, versus things with consistency are likely more of just a flag for you to check. All of these results should be listed within the NSDRG, whether they are false positives, true errors, or limitations of the implementation guide and your study, how they would interact. This validator report is not just for send ashore services. This is actually included with all of our send deliverables as well. Details about which rules are checked are listed in the additional tabs, along with their documented references. So a specific reference to the technical conformance guide or business rule, they are cited. Additionally, how each finding was checked is going to be included. So for example, if there's an identified duplicate record, we will include in that report that the keys that were used to define uniqueness in the define XML, so that you would either have to check your send data set, or we would for the send ashore, or if you need to define more uniqueness within the define.xml. Next, we're going to go into the quantitative and qualitative reconciliation report. Our reconciliation process, which is what sets PointCross apart really with this service, checks each data point across any source data that exists. So I'll give you a moment to sit with that. Within the PDF report, the tables and figures are going to be digitized. The send data is then going to be used to recreate the now digitized elements of the final report. These could include summary tables and selected groups that may have been included in the results portion of the report, pointing out some of these very important toxicology findings. With having the send and the tables and figures now digitized and being compared, the mean, standard deviation, standard error, and count are compared. If there is a mismatch in at least one of these elements, there is going to be a not matched flag that is presented in this report. This means that one of those items did not match, and it then has to be reviewed to make sure that they are either correct or there is an error in either the source data or the send data set. The qualitative data is recreated through a tabular format, and this is going to be checked by the presence of values. So while you cannot use numeric values, it is going to be checked by the presence and then a count of the presence of a yes or no or a count of severities. This is going to be automated where possible, and if not, there will be manual inspection. Any other data that is presented within the send data set that cannot be done through quantitative or qualitative tabular formats is going to be visually inspected through the XBiome graphical interface. Any mismatches that are reported within the reconciliation process are going to be added into the send to shore report. As a final reviewer, I typically hope that there are going to be no mismatches, but it does surprise me that some otherwise pretty good studies, while going through a review, have a few mismatches in count due to exclusion flags or slight rounding errors. As mentioned, these are all going to go into the send to shore report if they are true errors, but if they can be discounted due to just slight rounding errors, you're rounding a 17.1 to 17.0, we will note that and it may not require any further intervention. And here's just a little bit of a diagram for our reconciliation process. On the left-hand side here, we do have that the study report is going to be categorized and digitized. So information for the trial summary is going to go into a tool to recreate the trial summary domain. Information for the trial design, so this is going to be the elements, the arms, etc., is going to go into another automation tool, and that is going to recreate the trial design. Those are then going to be compared between the report and the send data. And then all other tables, images, etc., are going to be extracted and transposed into different tables. So we are going to call this the SRR, so the study report summary files. These are separated out into quantitative and qualitative summaries. So these summaries are then going to be extracted, labeled, and using the labels on each one of these, we are then going to transform the send data set, not actually changing the send data set, but pivoting it in certain ways to create the same results. If the mean, standard deviation, and count match, then the individual data points will match. If one of these values is off by any more than a slight percentage, then there's likely an error in the data set or the summary table. And then finally, I'd like to review what the send to shore report actually entails. This is going to be the main deliverable when we're offering this send to shore service. The send to shore report is a spreadsheet that reports all discrepancies, validation errors, warnings, and other findings in a tabular format. The spreadsheet is categorized into 3 severities. The first tab is going to be findings that have to be changed. These findings are going to be cited based on conflicting evidence, whether it is cited from a standards documentation, conformance rules, differences within the final report, etc. The second tab is going to be a should be documented. These may be things that are already documented in the NSDRG, but we're still going to include them. These are going to be items that produce an error that are found to be incorrect, but there may be no right way to fix them. Most of the time, this is a limitation as to how the implementation guide recommends a complex study be recorded, but the conformance rules might cause a conflict. And then you have an error as to you have to decide whether you have to abide by the conformance rules or the implementation guide. Um, Complex studies, this is fairly common to have to explain them. The third tab is going to be suggestions. These items are not necessarily right or wrong to include or exclude, but the information is present to populate or change the information within the SEND dataset. This might be something like including a principal investigator or a test site within a findings domain. And then I really do apologize for how small this is, but I think that gives you a little bit of information as to how we do prepare these. On the left-hand side, we're going to have, this is an example of a send to shore report. On the left-hand side, we do have issue types and where it's being presented, along with variable names, a description of the issue, and then the correction that is suggested and where it is going to be cited. Is this from a report reference? Is it from the send implementation guide? Or is this from a validation report? So the top two lines here are going to be missing parameters from the TS domain. There are 5 of them. There is one that is indicated as S length is missing, so the study length, and that is going to be coming from an FDA rule. It's labeled as an FDA compliance rule, and that is coming from the eData validator report. Pointed that out. There is also going to be data modeling issues. It's the second line here. And so it's saying that the parameters of PPTCNAM, PPTEGID, PPTEGSYM, and PPTMDA. These are all planned, the target common name, the pharmacologic target gene symbol, etc. These are all missing, but they are being referenced from the technical conformance guide and identified as missing within the TS domain. These are something, it's not in the implementation guide, there's not necessarily business rules, but again, it is in the conformance guide. And then the last line is saying that the PCDY and PCDTC are missing within the SEND dataset. These are going to be required to be populated for the reason of calculations. There were calculations performed in this study based on that, but these days or dates were missing in the SEND dataset entirely. For the second tab, items that should be documented, we will report any excess warnings and errors in this tab. Many of these findings are false positive warnings. There are some fairly common ones that trigger, such as consistency of the origin in the subject identifier. This warning is always going to trigger if you use pool ID in any domain, and it should be documented. The place to put any of these warnings is going to be in the NSDRG or the defined file, depending on the warning's details. If you're ever in doubt about something, please put it in the NSDRG. With Send Ashore, you'll see cost and time benefits. Any deliverables just involve time, typically time that a non-clinical researcher doesn't have, nor the expertise in Send. The Send Ashore process will provide you with a full detailed report about the conformance of your dataset at approximately 50% less of getting the full Send dataset from other preparers. And with that, and PointCross producing our own validator, as well as the creation and revision software, we can guarantee 0 rejections and 100% reconciliation of your dataset, allowing you to submit your study with confidence. And with that, I'd like to thank you for attending today. If you have any questions, again, you can put them in chat. We will answer any of them on our website, and you will get an email when those are posted along with the recording. You can also contact us at ask@pointcross.com. And again, the send generator tool is going to be on the main page of our website, and that's at pointcrosslifesciences.com. You'd be able to type in pointcross.com as well. I will be online for a few more minutes, so you can feel free to ask any questions. And if you want to direct any questions directly towards me, my email is cassandra@pointcross.com. Thank you.